Another question. So in a segment of DNA, they've given what total nitrogen bases have been given is 4500. Okay, now they're asking how many sugar molecules will be present. How many sugar molecules will be present in one strand of this DNA segment? Okay, this is the next question. Okay, we all know that the number of so here one thing to notice, in one strand of DNA segment they're asking how many sugar molecules are there. So we know that number of nitrogen bases in two strands, in both the strands, that is entire DNA, you have 4500 nitrogen bases, yeah, is equal to 4500. Now tell me the number of nitrogen bases in one strand, which is equal to 2250. Just divide by 2. Now we all know that the number of nitrogen bases is equal to the number of sugar molecules. Yes. So for that, let's have a look at this diagram. So this entirely forms a nucleotide. Yeah. So this is your nitrogen base. Sorry. This is your nitrogen base. And this is your sugar. So you know that number of nitrogen bases would equal sugar. Yes. So that is what I have explained, that number of nitrogen bases is equal to number of sugar molecule. Therefore, number of sugar molecules in one strand would be 2250. Okay, for this you just need to know the structure of a nucleotide. Okay, let's say this is your adenine. So this is your nitrogen base, this is your sugar, and this is your phosphate. So number of sugar molecules is equal to number of nitrogen bases. Okay, now let us move on to more complicated questions. Third one is number of thymine residues is in 5000 kilobases DNA containing 23% guanine residues is how much? So number of thymine residues in 5000 kilobases DNA containing 23% guanine is how much? This is very similar to the first question which we had solved. Okay, let's see. So here we know that A plus T plus G plus C is equal to 5 into 10 to the power 6 bases. So whenever they give you in kilo bases, you convert to bases. That's the first step. Now you do is 2 times T plus G, because they're asking the number of thymine residues. You're converting adenine in terms of thymine is equal to 5 into 10 power 6. Now T plus G is equal to 2.5 into 10 power 6. And because 23% of guanine they have given, so you know that how do you convert this percentage? So in 100 bases you'll have 23 bases as guanine. Now the question is how much bases would be guanine in 5000 kilo bases, that is 5000 into 10 power 3 total bases. How much would be the number of guanine? It would be 5 into 10 power 6 multiplied by 23 divided by 100, which is equal to 115 multiplied by 10 power 4. Yeah, basically you're converting this percentage to this. So this is the trick over here, the tricky part. So now you can write as 2.5 into 10 power 6 minus 115 into 10 power 4, or basically 250 minus 115 into 10 power 4, which gives you 135 into 10 power 4, which in standard notation you'll give us 1.35 into 10 power 6. So this is the third type of question which you can solve. So that's it, and I hope I covered most of the tricky part here. So if you have any questions, comment.